Welcome students. In today's lab we are going to talk about the histology of the skin. The skin is formed of two layers. An epithelial layer superficially called the epidermis, and beneath that, below that, deep to it, we have a connective tissue layer called the dermis. Both of these form the skin. So here we have, this is the epidermis, appears dark stained, and the lighter stained red in color, mostly red in color, this is the dermis. Below the skin, we have a third layer called the hypodermis, or the subcutaneous layer. This is mostly formed of fatty tissue, and therefore, meaning that it is mostly we have adipocytes, and as you know, these adipocytes appear white or vacant in color. So as you can see in all these parts, the cells appear white or vacant in color because mostly it is fat. Now we start with the epidermis. The epidermis is an epithelial layer. It is a stratified, squamous, keratinized epithelium. Before we get into the details of the epidermis, let's look at the epidermal-dermal junction. Notice here, this junction, it is not a straight line. It is not a straight line. It is characterized, it is irregular and characterized by the interdigitation of epidermal ridges and dermal papillae. The epidermal ridges, as you know, are downward projection of epidermal tissue. These are all epidermal ridges. And dermal papillae are upward projection of the dermal tissue. And these interlock with each other, they interdigitate with each other to form this irregular jigsaw-like junction. Now, sometimes, especially in thicker skin, this is even seen, this is even reflected on the surface of the skin. Notice here the surface. Here we have a series of ups and downs on the surface. These are called also epidermal ridges. Now these epidermal ridges on the surface with the grooves, the ridges here and the grooves here, these form the basis of the fingerprints. So let's look at the layers of the epidermis. First, here we have a layer, the deepest layer here. This is the stratum basale, stratum basale. Then after it, we have this part here. This is the stratum spinosum. And then we have a narrow layer formed of only 3 to 5 sublayers. This is the stratum granulosum. This pale staining thin area, this is the stratum lucidum. And finally, this thick region, this is the stratum corneum. This is the stratum corneum, the most superficial layer. As you can see here, we have 5 layers: basale, spinosum, granulosum, lucidum, and corneum. And as we have said, if the epidermis is formed of 5 layers, this is classified as thick skin. Classified as thick skin. Again, here we can see the ups and downs on the surface of the skin. These are the bases of the fingerprints. Let's look at these layers. Starting with the stratum basale. This is the deepest layer. It is formed of a single layer of cells. It is only a single layer, one layer of cells. Now, it is highly mitotic It's not easy to see that in the light microscope. And here, in addition to the keratinocytes, we have Merkel cells and we have melanocytes. These large pale staining cells, these are the melanocytes. These are the melanocytes. And actually, melanocytes are the only cells that are the only non-keratinocyte cells of the epidermis that can be easily seen in the light microscope. It is not easy to see the Merkel cells or Langerhans cells. The second layer is the stratum spinosum. Stratum spinosum is formed of several sublayers. It is usually thick, and it is thicker and thicker skin. When we prepare the tissue for histological study, the cell cytoplasm will shrink and the tonofibrils will produce the spine-like structures on the surface of the cell. So in this image here we have a cell and we have these spine-like projection on the surface. These are caused by the tonofibrils, which do not shrink, of course, like the cytoplasm. Then we have the stratum granulosum. Stratum granulosum is formed of about 3 to 5 sublayers. This is the stratum granulosum. If you look at the cells here, the cells here are characterized by the presence of darkly stained granules. These are the keratohyalin granules that we talked about, which appear as darkly stained structures under the light microscope. Then, superficial to it, we have the stratum lucidum. Stratum lucidum, a pale staining, thin region found only in thick skin. Found only in the thick skin. Finally, the most superficial layer is the stratum corneum. All this is the stratum corneum, and it is thick layer and thicker and thicker skin, and it is formed of many sublayers. And the cells here are flattened squamous cells. Of course, they have no nuclei and no organelles, and they are nothing more than bags filled with keratin. If we examine this epidermis, the darkly stained layer here, the deepest one, this is the stratum basale, and then we have the stratum spinosum, this is the stratum spinosum, and then here we have the stratum granulosum, this is the stratum granulosum, and the most superficial layer is the stratum corneum. As you can see, we don't have stratum lucidum here. We don't have stratum lucidum. This means that this is a section through thin skin. So in thin skin, the epidermis is formed of only 4 layers, and we don't have the stratum lucidum, and in addition, it is thinner. The epidermis is thinner than in the case of thick skin. Now the dermis, the connective tissue part of the skin. The dermis is formed of 2 regions. The papillary region, this is the most superficial part. It corresponds to the dermal papillae. So this is the general area. This is the general area of the papillary region. So this is papillary dermis, all this is papillary dermis. It's formed of loose areolar connective tissue. Now, deeper to it, we have the reticular region. The reticular region. all the rest, all this is the reticular region. All this is the reticular region. Okay, this is the papillary, and all the rest is the reticular. This is the deeper part of the dermis. It is formed of dense, irregular collagenous connective tissue, and most of the other structures of the skin are found here, from the hair follicle to the glands, the arctopili muscle, and to many sensory receptors. We're going to talk about only two types of sensory receptors in the dermis. First, here, if you look at this image, here we have the epidermis, and here we have the dermis, and this is the junction between them. As we have seen earlier, as you can see here, it is irregular. Here we have an epidermal ridge, and this is a dermal papilla. If you look at in this dermal papilla, we have this elliptical structure that is perpendicular to the direction of the epidermis, and this is Meissner's corpuscle. Deep in the reticular dermis, and sometimes even in the hypodermis, we have the lamellated or Pacinian corpuscle. Usually, we should have a nerve in the middle, surrounded by several concentric layers of flattened Schwann cell, which are surrounded by several concentric layers of connective tissue, and then all the structure is surrounded by a capsule. And this appearance is called onion-shaped appearance of Pacinian corpuscle. Pacinian corpuscle. Here also on the slide we can see some sweaty glands. Now the darker structures are the ducts. These are the ducts. And the lighter structures are the secretory portion. So these are ducts, and these are secretory portion. Now, the hair. This is a cross section through a hair root, and if you look at the parts of the hair, in the exact middle, in this region here, we have the medulla. The medulla is surrounded by the cortex. The cortex is formed of cells that have darkly stained dots. So as you can see, these regions, in this region here, we have several dark stained dots, and this is the cortex. The cuticle appears as a very thin pinkish line surrounding the cortex. This is the cuticle of the hair. A thin pinkish line that surrounds the cortex. Now these are the parts of the hair. What about the hair follicle? The hair follicle is formed first of all of an epithelial root sheath. Epithelial root sheath formed from cells that are derived from the epidermis. As you know from your lecture, it is formed of internal and external root sheath, but we are not going to go into these details in the lab. So, the cellular part here, the first part of the hair follicle, this part here, is the epithelial root sheath. Outside the epithelial root sheath, we have the dermal root sheath, formed of connective tissue derived from the dermis. This part here, connective tissue, dermal root sheath. Between them, we have the thick basement membrane, this red line, this is the glassy membrane, which is the thick basement membrane between the epithelial root sheath and the dermal root sheath. If we look at this longitudinal section through the hair, notice here we have the epidermis. This is the epidermis. Now here we have the hair follicle. This is the hair follicle. Notice how the hair follicle is a downward projection of the epidermis. So it is derived from the epidermis. The hair follicle is derived from the epidermis. Associated with the hair, we have sebaceous glands. This is a sebaceous gland, and this is sebaceous gland. And we have the erector pili muscle. For example, this here, in this part here, this is the erector pili muscle. As you can see, we can barely see in this part here and this part here. These are hairs. These are found outside the skin. This part of the hair is found outside the skin. So this is shaft. This is shaft. So we'll take the same image and magnify it. So this is a magnified view of the previous slide. Here is the hair follicle. This is the hair follicle. And here we have the erector pili muscle. Erector pili muscle. This is the erector pili muscle attached to the dermal root sheath. This is the dermal root sheath. Notice how this is the sebaceous gland. Notice the position. The sebaceous gland is located in the angle between the hair follicle and the erector pili muscle. Also, note the color of the cells. The cells of these glands appear clear or white because the secretion of these cells is mostly formed of lipids, and the lipid disappears during preparation, and therefore these cells have this appearance. Again, here we have some sweaty glands. Sweaty glands. The darker structures are the duct, and the lighter structures are the secretory portion. Also, here we have sweaty glands. Here, these are also sweaty glands. And by this we conclude our lab for today. Thank you and good luck.